This is the second video in the series called Enzymes and Metabolism. In the first video I introduced enzymes and gave some background which is pretty important to understanding some of the things that go on in this one. So if you didn't get a chance to watch that video, I'll put a link up here at the end of this one. You can go back and have a look at it. But for the time being, if you did, you'll recognize this. And you'll know that we're talking about enzymes and substrates and products and that sort of thing. Enzymes, if you remember, they're very large polypeptides. Maltase, for example, has over 900 amino acids in it. Maltase can be extracted from liver tissue, 900 amino acids. So if you think about it, you take 900 amino acids and you make an alpha helix and you put some proline in it and it bends and folds and does all this kind of stuff. This could be an enzyme. Huge, huge molecules. So big, in fact, that they can actually be seen under electron microscopes. You can see them, you can see where they are. You can't see the detail of their structure, but you can certainly see maltase as a huge, huge molecule. We know it does things like this. If this is maltase, maltose, forming an enzyme-substrate complex, maltase, and glucose, two glucose molecules, that's what that would represent. In this video, we're going to take a closer look at this relationship. But before we do, we have to recognize that because you can't see what's going on here, it's big, you can't actually see it, people have developed models to try and explain how it works. This is an abbreviation of something that's called the lock and key model. And if you think about locks and keys fit into a lock and allow something to happen. If you take a different key, it may fit into the lock, but it won't necessarily unlock anything. The reason it's called the lock and key model is because of the absolute uniqueness of shape and how important shape is. And if the shape isn't exactly perfect, this key might work, this key might not. And they're very, very similar. Lock and key model. So it looks like this. Here's our enzyme, and what I said before is enzymes have a region on them, which is, we know it's called an active site. And in this one, I'm just going to color this piece in right here. And I'm going to call that blue region right here the active site. That's the portion of the enzyme which will actually interact with the substrate. So in this reaction we've got maltase, this is our maltase, and in the abbreviation this is maltase, combines with the substrate, which in this case is going to be maltose. So we have a maltose molecule. Let's suggest that this is a maltose molecule. And you can see that the shape of this is such that it can fit into that space, into the active site. That's the idea here. The shape of these things is so critical. So I'm going to say, okay, we've got a bit of a, I'm going to draw this again here. It doesn't look exactly the same, but it's close. Close enough for what we're doing here. And if that's the enzyme, the substrate, maltose, this is our enzyme, fits exactly in there, into the active site. And it's sort of like presto changeo, something happens and you get a product out of the thing. This, I'll just color the active site here. It's still there. This thing here is known as the activated enzyme-substrate complex, the ES complex. What I mentioned before is that the active site is actually, I'm going to represent that down here again. So this could be the active site on the enzyme. The active site is lined with amino acids, and these amino acids have got There are groups which are exposed into the surface, onto the surface, into the region where the maltose molecule can combine. Maltose itself, as a molecule, has hydroxide ions, their hydroxide groups are sticking out for it, so it has certain polarities and certain chemical nature to it. This thing is so precise that it's the chemical nature of the molecules that interact that actually allows this thing to get in here. So there's an interaction in here and it's held in there momentarily. How the reaction occurs is not part of the lock and key model, the lock and key theory. I should have written that up here. Lock and key model to explain enzyme activity. But the actual theory, the belief, the hypothesis as to how these things actually work is called the Induced Fit Hypothesis. The lock and key model, the origins of this idea that enzymes and their substrates work like locks and keys are that precise, that the origins of that model are in the late 1800s, like 1890. That's an old idea. Electron microscopes weren't invented until the 1930s, and nobody even saw enzymes. So since that lock and key model was developed, they've added to it, they've added this induced hypothesis, induced fit hypothesis. And what it actually says is that when these two chemical species get closer to each other, they actually force a slight change. So as maltose gets in here and bonds temporarily in this region in the active site, it forces a bit of a change to the enzyme, and that's what it says. It's inducing a fit. It induces a fit so that it actually tweaks the shape a little bit, and it's believed that tweaking that shape causes the reaction. The enzyme's not going to change much. The enzyme is a great big thing and is held together by a multitude of bonds, but maltose, which is two sugar molecules bonded together, actually will break apart as a result of that. So we wind up with our products. These are glucose. The enzymes right there. Notice a couple things. The enzyme isn't changed. The enzyme remains. The only thing that really changes is maltose becomes glucose. The enzyme is left so it can react again. Cells are pretty efficient when it comes to things like this, but they don't make an enzyme every time they need to have a reaction. They use the same enzyme over and over and over again. So realize that the enzyme is there to be used again. And what I had before, if you think for a second, let me just get rid of that. We don't need the words either. Is that these reactions are reversible. So that if we're going this way, this is our hydrolysis reaction. If we go that way, that's dehydration synthesis, or condensation as some people call it. If we're going this way, water's given off. If we're going this way, we have to have water present so that the hydroxide ion and the hydrogen ion can go back onto the glucose molecules when the maltose is taken apart. The point is, this is always present. So it's there should the reaction go that way, or it's there should the reaction go this way. It's so important to realize the significance of the shape. That's critical. Enzymes work when they're the correct shape. That's it. If you do anything to a system that changes the shape of that active site, then that substrate won't fit. If you do anything at all to change the shape of the key or the lock, whatever we're talking about, the shape won't match and no reaction is going to occur. It's so important to recognize that. the things that can affect the shape of these is really the topic in the next video. There's a number of conditions which can affect the shape of enzymes and therefore their function, but that's the topic of the next video rather than getting into that here. I do want to extend this just a little bit though, because it is possible, let me erase some of the extra stuff here. Let me just erase this part. It is possible to have another molecule that's very similar to the substrate. For example, maltose is a disaccharide, we know. Well, there's another disaccharide that we know, and it could be sucrose. Very similar in shape because this is glucose plus glucose, this is glucose plus fructose. So they're very similar in shape. It is possible to have a molecule that is so similar in shape that it will fit into the active site. It's not going to work. It's not the correct fit. It'll get in there. You can take a key and you can jam it into a lock and it won't unlock it. That's what this is. You can have something that will fit but will not unlock it. These things are called competitive inhibitors. Inhibit. So competitive inhibitor is something that can fit into the active site. It'll occupy the active site and it'll actually block the reaction. It's called competitive because it competes for occupancy in the active site. Inhibitor because it'll block the reaction. So it's really a concentration dependent thing because in a system, if you've got so many enzymes, you've got so many correct substrates and you've got a bunch of inhibitors, some of the enzymes could be blocked, some of them might not be blocked. So it's really a concentration thing, how many of each there are and how effective they are. So competitive inhibitors, the presence of those is one thing that does affect not the enzyme shape so much, but it'll affect the ability of the enzyme to function. There's something else that enzymes need as well. They also need cofactors. So this is something that can block it. Cofactors are often required. You say, okay, so what's a cofactor? Well, cofactors can be either of two things. They can be mineral ions. So we have things like calcium is required in our bodies, right? We have things like sodium is required in our bodies, and things like that. These are all minerals. Or you could have what we call vitamins. Now, vitamins are large organic complexes. They're also called coenzymes. Now what these things do, these cofactors, is they help stabilize electron distribution. So there's going to be a reaction, and these things are required in order to help that. Because in any time you've got these things breaking apart or joining together, there's electrons being moved, there's bonds being broken, and you can't have big shifts in those things in a system that's supposed to be living. So it doesn't, these things help in that way. Calcium, sodium, I can give you an example. This one is large organic complexes. This example is going to be used in the fourth video in this series. Niacin, in the form of nicotinamide adenine dinucleotide, NAD. It's part of the B vitamins. So you often think, okay, what do we need all these vitamins for? Why do you need to build strong bones and teeth? Yeah, but how? Because there's chemical reactions that happen, or enzymatic reactions that happen, and these things assist in those reactions. Now, just before we leave this topic and leave the topic and get it ready for the third video, I want to introduce one more little bit here. There's a set of terminologies that is used in this topic. The apoenzyme is a term that just means the protein portion of the enzyme. There's a thing called the haloenzyme, which is the protein portion plus the cofactors. Because what we really have, and I'm just going to take this bit away here, what we really have is an enzyme such as that one, where it's got an active site, but unless there's the correct cofactor associated with it, it's not going to work. So you have the enzyme and you have a cofactor, whether it's a vitamin, or whether it's just an ion, but there's the combination of these things, which is the haloenzyme, that really forms the activated enzyme-substrate complex. So, I think that's it for this one. The next video is all about what kinds of things change the shape of this, and what's the effect of that on the chemical, on the enzymatic reactions. Remember, if you want to go back and have a look at the previous video, it'll help you with a basic understanding of what this is about. There's your link for that, and my gecko for you if you'd like to subscribe. Thank you.